Hello, everyone. Today's lecture is going to be on one of my favorite topics, plant genetics and plant breeding. I'm Dr. Wood, and here's the lecture. So, the outline of today's lecture, first, I'll be going over Gregor Mendel and his studies on inheritance in peas, and then I will discuss some classical plant breeding techniques, and then I will go into molecular genetics and DNA. And then I will touch briefly on molecular plant breeding. And this first section will be on Mendel and Mendelian genetics. And I like to start with Mendel because genetics was actually discovered by Mendel using plants. And Gregor Mendel was an Austrian monk and an avid gardener. He lived in the 1800s, and he was interested in the inheritance of traits. So offspring resembled their parents with some differences, but why was that? So a few definitions here. A trait is basically any specific, obvious, observable, and measurable characteristic of an organism. And so the 7 traits that he looked at in his study were stem length, pod shape, seed color, flower position, flower color, and pod color. So those are the different traits. And then for each trait, he saw 2 different types. And nowadays we call these phenotypes. So those are the physical characteristics, the physical manifestation of the traits. And you can kind of think of them as like the different options that a trait can have or different flavors. So all plants have stems, but some are going to be tall and some are going to be dwarf, okay? Or like yellow and green for the seed color. So those are the different phenotypes is the term we're going to use. And so Mendel lived before the invention of genetics. He's pretty much the one who discovered it. So how did he do genetics without knowing anything about DNA? Well, basically his raw materials were a bunch of pea plants, paintbrushes, a lot of spare time, a really big garden, and he liked to count things. So what he did is he basically crossed the pea plants. And the method he used is demonstrated here. So basically, you would cut off, so one of the plants would be the female parent and one would be the male parent. So this is the symbol for female. He would cut off the stamens from the plant that was going to be the female parent. And if you remember from flower anatomy, the stamens are the male parts. So he'd cut those off before they were mature, so that way the pea couldn't self-pollinate. And then he would take his paintbrush and he would pick up pollen from the stamens of a male plant that had a different phenotype. And he would take that and he would basically paint the pollen onto the stigma of the female parent, and then the pollen would go and fertilize the egg, resulting in seeds in the pea pod. And then he would take those pea pods and plant them out. I read somewhere that he grew over 28,000 plants over 8 years for his experiments. So he grew them out, and then he would count the number of traits he saw. And this was also before the invention of calculators. Anyway, so he did something called a monohybrid cross. So basically looking at one trait, so mono means one trait at a time, he would cross the plants with different phenotypes and observe the hybrids. And okay, let's walk through Mendel's experiment. So in this case, we're looking at 2 parents that differ by flower color. So one has the purple phenotype and one has the white phenotype. So he took the pollen from the white one and pollinated the purple one, grew out, collected the seeds from that cross, and then he grew out a bunch of plants, and he saw that they all had purple flowers. So 100% of that next generation from the parents had purple flowers, and so this is called the hybrid generation or also the F1 generation. And then for the next step, he put away his scissors and paintbrush because he just let these hybrids self-pollinate, which pea plants do naturally. So these all self-fertilized, self-pollinated, and he collected all the pea pods from those plants and grew them out for the F2 generation. And what did he see? So he looked at almost 900 different F2 plants, and he saw a mixture of purple and white flowers. Although you could see that there were more purple flowers than white, or rather plants that have purple flowers than white. So yeah, in the second generation, the F2 generation, most of the plants are purple, but the white flower phenotype reappeared. And this is the ratio that he got in his study. So yeah, 700 plants had purple flowers, and 224 plants had white flowers, which is a 3-to-1 ratio. Another way to represent that is it's 75% purple and 25% white. But Mendel did not stop there. He decided to take it to another generation, so the F3 generation. So he took, for example, a white flower and let it self-pollinate, and he found that those white flowers had 100% offspring with white flowers. And then he would take a purple flower from that F2 population, self-pollinate it, and some of the purple flower plants from the F2, their offspring had 100% purple flowers. But an interesting thing is that some of the purple flowers, even though they look the same as the other purple flowers, they had offspring that had 75% purple flowers and 25% white flowers, just like the generation before in this pie chart. So he drew a couple conclusions here. First of all, the purple phenotype is dominant, so it masks the appearance of the white phenotype. And the white phenotype was said to be recessive, and he called it that because basically it disappears for a while in one generation and then reappears, so it kind of like recedes, but then it comes back later. In F1 hybrids, they only show the dominant phenotype, but then the recessive phenotype reappears. So you can remember recessive reappears. And yeah, he didn't just do that for flower color. He did it for 7 different traits shown here. And all of them showed the same thing, which was the F1 hybrids, where they all showed the dominant phenotype. So 100% dominant phenotype in the F1. And then in the F2 generation, he saw 75% showing the dominant phenotype, just like with the purple flowers. So he thought this math was really interesting, and kind of the ratios especially gave a hint as to what was going on underneath the surface. So even though Mendel didn't know about genetics, because he hadn't invented it yet, and there wasn't really an understanding in the time of meiosis either, they did understand that the egg cells in the female parts of the plant and the pollen cells in the male parts, that those were the reproductive part. But he kind of, from his data and the way that the ratios came out, he basically hypothesized there must be kind of like 2 things in the parents that are getting randomly arranged into the offspring. And so here's what is, and it ended up being right. This is what's going on beneath the surface here. So basically the purple parent, the female plant, has 2 copies of something for purple, and the white has like 2 copies of the white character. And then these different copies get randomly put into the reproductive cells, the gametes. In this case, they're all going to be the same as the parent, because the parents are homozygous. And homo just means same, so they have the same alleles in this case. Anyway, when the egg cell joins with the sperm cell during fertilization, that forms the zygote, which eventually will become the embryo, and then eventually will become a full plant. And this plant will have 1 copy from one parent and 1 copy from the other parent, so it's said to be heterozygous. So that's the hybrid. And you can see it's pretty obvious how that plays out. This is a Punnett square here. This little square guy. You get the way that you read these is basically you take the two options for gametes from each parent. In this case, they're the same for both parents, and then you give the zygote one from the female and one from the male, and that's what it's going to have. Okay, so then, all right, so let's take it to the next generation. So in an F1 by F1 cross, where you have two hybrids that you're crossing, or it could be also the same hybrid that's self-pollinating, where it's the same flower it's acting as the male and female parent. It's going to be the same result here. You basically, what you get is each hybrid has one purple and one white from each parent, and those are going to get randomly assorted into the gametes. So half of the egg cells will have the purple and half will have the white, and then same with the pollen. So then what happens when the egg and pollen come together to make the zygote? So here you can use the Punnett square to figure that out. Basically, if the purple egg meets with the purple pollen, then you get a zygote that has two purples, and so the plant's going to be purple. If you get a white egg cell with a purple pollen, then your zygote will have one white and white purple, so it'll appear purple, have the purple phenotype, but it will still be a hybrid genetically. Same in the case where it's a purple egg and a white pollen, and then if you get the white egg and white pollen, then you get a zygote that has two whites, and that one will be white. And this is what he saw. So this is what he came up with to fit his data, which was he was seeing 75% purple flowers, 25% white flowers. So he reasoned that, that each parent must have kind of these two possible versions that where one is dominant to the other. And that's how he came up with this theory. And all right, and here are a couple useful definitions for understanding Mendelian genetics. So the first one is allele, and so you can think of that as a particular variant of a gene or different flavors of the gene that can influence the phenotype. So like the purple allele and the white allele lead to the purple and white flower phenotype. And dominant alleles mask the presence of recessive alleles. If an individual has two identical copies of the same allele for a given gene, it's said to be homozygous. And if it has two different alleles at that same locus, then it's heterozygous. And so the options you have for Mendelian genotypes is you can have homozygous dominant, homozygous recessive, or you can have a heterozygous, like a hybrid. And so from these experiments that Mendel did, he came up with his three principles. The first one's dominance, so one parental character will dominate the other in the F1 generation. Segregation, the dominant and recessive character separate out in the F2 generation here. And it also kind of refers to how the two alleles segregate randomly into the gametes. And finally, independent assortment. So the parental characters for different traits, so the parental alleles basically for different traits, will segregate independently of each other, regardless of each parent they came from. So, and he figured that one out here. And so he figured out the law of independent assortment by doing these dihybrid crosses. So basically he took two parents that differed in two traits instead of just one. So like for example here, one parent would have purple flowers and yellow seeds, and the other parent would have white flowers and green seeds, and then he crossed those. And at first it looks pretty similar to what he saw just for one trait. So the F1 hybrid, in this case, they all looked like one of the parents. both of the traits. But in the F2 generation, you got to see, he saw actually new combinations of flower color and seed coat. And he did this for all of his different traits. That's why he grew so many plants, and it took him 8 years. But yeah, he figured out somehow that these different phenotypes are segregating independently in the F2 generation. And Mendel didn't know about DNA or chromosomes or meiosis, but the reason we know now is it's because the traits that he picked, the genes that control those, are on different chromosomes. And we know now that chromosomes segregate independently into their gametes during meiosis. So that explains why independent assortment happens. But pretty neat he was able to find this out so long ago. Okay, and it's worth mentioning that there were a couple things that made Mendel's peas especially successful for figuring out genetics. And for one, the first thing that he did was start with a really diverse collection of peas. So if you read his paper, which I did to prepare for this lecture for some reason, he says, in all, 34 more or less distinctive varieties of peas were obtained from several seedsmen. So he went around and gathered peas that are different from each other, because you can't really do genetics if you don't have any differences that you're trying to study, right? And then he had the variation in phenotype, and it was like heritable. He's trying to study inheritance, right? He was pretty lucky that peas are diploid, so they only have 2 copies of each gene instead of having more like some plants do. That they reproduce sexually, and that they're self-compatible. Those were all important for his experiments to work. And then something that he did, which helped a lot, is that he took those diverse lines and he inbred those lines for 2 years before starting his experiments, before starting the crosses. So he just crossed them to themselves, and he really wanted to see a stabilization of traits. He wanted them to be really uniform. So like all of the whatever flowers would be purple on one of the varieties he was interested in, for example. And then this was really just a stroke of luck, or maybe he did this on purpose. I don't know. But basically all of the all of the genes underlying his traits happen to be unlinked in the pea genome. So those genes were on different chromosomes. So that's why he got independent assortment. It might have been confusing if he saw something else. And another thing to note, even though he was really successful and did great work studying genetics using only peas and math and time, his note was actually completely ignored during his lifetime. He stopped doing genetics research after the pea experiment paper was published. He tried to reproduce it in another species, but that species actually reproduced asexually in the flowers, and he didn't know that at the time, so his results were not looking very good. So he kind of gave up on research. Luckily, his work was rediscovered in the early 1900s, and was really important for coming up with the field of genetics. So we like to give him homage in the modern times, even though he was underappreciated in his days. And Mendelian genetics, it was pretty much spot on for how genetics works, although of course it's going to be more complicated than that in real life, and there have been a lot of things discovered since then that build on the work, but explain other phenotypes that we see. The first one is incomplete dominance. So in incomplete dominance, you see an intermediate phenotype for the heterozygous condition instead of seeing a dominant. And I'll show a picture of that in the next slide. Also, some traits have multiple alleles, so more than 2. And the common example is blood type for that in humans. Another thing is that polygenic inheritance. So sometimes multiple genes will influence a trait. Like multiple genes in different locations in the genome will determine things like color and height, etc. And it turns out that the Mendelian math and ratios still work out for those in a lot of interesting ways. And finally, as mentioned, linkage. So his traits were unlinked, but a lot of genes and traits are linked, which means they live on the same chromosome and they get inherited together. And so if two genes are links, they're not going to do the independent assortment thing. So they're going to be usually inherited together. And I'll talk a little bit more about that later. And here's the example, one example of a non-Mendelian genetic system of incomplete dominance. And this is pretty common. So if you cross two flowers, one's red and one's white, and you see that the F1 generation is pink, so somewhere in between the two parents, then you're looking at incomplete dominance here. But it still segregates the same way as Mendel's peas. So when you cross that F1 generation, you cross those hybrids, you see this ratio. It's 1 red, 2 pink, and 1 white. So, and this is explained by the genotypes. So the red plants are going to have 2 of the dominant allele are. The pink plants are heterozygous, so they have 1 big R, 1 little r. And usually with these Punnett squares and other things, the capital letter is the dominant trait, although in this case it's incompletely dominant, and then the lowercase letters are the recessive trait. And then my face is covering it here, but the white flower is going to have 2 lowercase r's. So that's where you get the 1 to 2 to 1 ratio. So that's the end of the Mendelian genetics section. And this section is going to be on classical plant breeding. And by classical, I just mean before the discovery of DNA. What techniques did people use to do plant breeding, and what were their results? And first, let's talk about the difference between natural versus artificial selection. So here we have Charles Darwin, and he's famous for his theory of natural selection and evolution. And so the idea here is that basically in nature, those who survive the best and have the most offspring in a certain environment will pass on their characteristics to the next generation. And then that would result in this gradual change of a species over time into a new species. And so that's evolution. And Charles Darwin was actually really inspired by animal husbandry, and the breeding of dogs, I think specifically. And so he saw that people were able to select traits they wanted in species and change them over time. And his big leap was realizing that nature could do that as well, and the environment could influence that. So natural selection, artificial selection are similar, but in artificial selection, humans are the ones who are controlling reproduction. So it's selective breeding, and humans have done this for a really long time. Important to note, both forms of selection are acting on traits. So they're selecting the best phenotypes. Okay? So in order for selection in either form to make a change, there must be phenotypic variation for selection to act upon. Otherwise, there's there would be no difference in phenotypes, so there would be no diversity. And also, so selection is acting on the traits, but the variation in the traits needs to be heritable. So there is a genetic basis, but selection isn't acting on genes directly. And I think that's important to note. And humans, selected for different traits for millennia, without knowing anything about genes. So here's a bunch of examples that I found on the internet. And we talked about this a little bit in the first lecture, but it's been a long time, so I thought I'd refresh your memory. Humans selected for bigger grains and bigger fruits, better taste, and more digestible products, easier to harvest and process, and also like more uniform, which also made it easier to harvest. And this first example, or this one on the bottom here, that's a wild relative of the watermelon. And here is this like Dutch painting from like the 1600s, and then you have the modern day watermelon. So you can see it got a lot bigger, a lot fewer seeds, and also the wild melons were pretty bitter, and the current ones are pretty sweet. Here you have a papaya, similar story, bigger, higher fruit or flesh to seed ratio, which is nice. You got teosinte and corn, and then this is a wild banana. It's also full of seeds. And then you have our modern banana here, which is seedless and a lot bigger. So, and we also talked about this in the first lecture, but basically crop domestication occurred all around the world on different crops, like all around 10,000 years ago or so. And that's where the beginnings of agriculture began. So, and basically this plant breeding, genetic modification by plant breeding started to happen because of cultivation. So basically, the early farmers would grow a crop. There'd be some natural variation within it. And then the good farmers would plant seeds from the good plants the next year. And so the traits of those good plants got carried on to the next generation, and then over time resulted more and more of the plants looking like the good parents, and that's how the early plant breeders did it. And here's another example with using corn. So it's corn, also known as maize, and so that was domesticated in Mexico by indigenous farmers around 10,000 years ago. And this is the wild relative of maize. It's called teosinte. And teosinte, it's kind of edible with some work. Each kernel is individually wrapped in a hard coating, so they had to be individually peeled. And each kernel is a lot smaller than a kernel of corn even. So it's a lot of work for not that much reward. And also their teosinte kernels are pretty much indigestible by humans on their own. And the indigenous people of Mexico got around this by inventing another technology, which was called nixtamalization. And so that's a treatment with calcium hydroxide, which is alkaline, and it's actually still used to make like masa for tamales and tortillas and such, and pozole, so, which is really delicious. But we don't have to do that anymore with corn because we bred it to be digestible by humans. And I think this picture here kind of shows that's the outer coating, how it has evolved over time to just be like kind of the bottom part of the corn kernel. And also in corn, the overall plant phenotype changed over time during domestication. So the ancient teosinte was really branchy, and the kernels were very small. And then in modern corn, you pretty much just have like one main branch, which is nice for the way that people do agriculture now. You can fit more plants in the field and like harvest a lot easier. And then the seeds are much bigger. Have to bring this up. So the traditional narrative of plant breeding and domestication is that humans over time manipulated the plants through the selective breeding practices. But there's a really great book by this Berkeley professor, Michael Pollan, Botany of Desire, and he argues that actually it was the plants that were manipulating the humans into spreading their genes by appealing to our desires. And anyway, it's a great read. There's also like a Netflix documentary made about it, made based on it. And he like goes over the history of these like four crops: apple, tulip, potato, and cannabis, and their relationship, and like how they were domesticated and bred and popularized in different ways, and their relation to different human desires. So highly recommend reading that. All right, and then I just wanted to mention a few famous plant breeders in case you haven't heard a lot about them. The first we have Norman Borlaug here standing in a field of his dwarf wheat that he invented. And so this variety, it's shorter, which means it uses less agricultural input. So less fertilizer, less water to get the same or even more yields than traditional wheat. Next we have George Scholl here, pictured sitting in his greenhouse, I think. And in the early 1900s, he invented hybrid corn and popularized that, which was also important for increasing agricultural productivity. And I'll show a picture of that in a later slide. And then finally we got Luther Burbank here, Santa Rosa's darling. He lived in Santa Rosa and kind of popularized plant breeding as a business that you can do. And he himself invented over 800 different cultivars of different crops and different plants. And probably most famously the Burbank potato, which is the quintessential Idaho potato. There is a museum in Santa Rosa where you can learn more about him and you can see his gardens. So definitely recommend checking that out if you want to learn more about him and his breeding. All right. Here I just wanted to talk a little bit about the difference between qualitative versus quantitative traits, which is important for plant breeding. So qualitative traits, that's all of the traits that Mendel looked at were qualitative traits. So basically they fall into discrete phenotypes. So they're easily distinguished from each other and they're different from each other. These tend to be determined by a single gene. All of Mendel's were. And they're not very influenced by the environment. So whatever gene, whatever allele a plant or organism has, that's like what phenotypes they'll get. Examples, all of Mendel's traits, so like flower color is a qualitative trait. You either have purple or white. Also stature, so, plant height can vary continuously. But in Mendel's case, he categorized it into dwarf or tall. And so the gene here, there was no kind of in between. It was either tall or it was dwarf. On the other hand, quantitative traits. These show a continuous range of variation. So you often see that depicted by this bell curve or normal distribution here. So on this chart here, you basically have the phenotype from in height. And so this is the number of individuals. So most individuals are in the middle, but there's some that are tall and some that are small. The plants would show a lot of in between. And quantitative traits tend to be determined by many genes. And their phenotype that they show for a quantitative trait, like height, how tall they get is going to be a product of their genes plus their environment. And some common examples of this is plant height, as we showed below, and crop yield, another one. So, like I just said, the environment has a big influence oftentimes on quantitative traits. So, for quantitative traits, the phenotype that you see is going to be a product of the genotype plus the environment. And an example of this from this class is the media trials that we did. So we measured plant height. So that's a quantitative trait. It varies continuously, quantitatively. And when we grew, this graph I think is corn. When we grew the corn in the different growing media, that significantly affected the plant height. So all of the corn that we grew, they were all there weren't any big genetic differences, right, in the corn seeds. But you did see really big differences in the phenotype, and that's because some of these media, the growing media, had more nutrients than the others, right? And so there's a concept in breeding known as heritability. And so heritability is basically how heritable a trait or a phenotype is. So it's what proportion of the phenotypic variation is due to the genes. And if you have a trait with low heritability, that means that the phenotype for that trait is mostly determined by the environment. And, for example, plant height and nutrition of soil. And then how do you deal with the influence of environment on plant phenotype during breeding? One way to do that is to grow them all the plants in the same environment. Or you can grow plants in different environments, but like all the plants you're testing should be tested in every environment, right? So I also wanted to show what breeding kind of looks like on quantitative traits and like the impact of selection. So let's see here. So like I said in the last slide, phenotype is genotype plus environment, and then heritability is what proportion of that variation is due to genes. And so there is this equation called the breeder's equation, which is just basically the response to selection that you're going to see. So how much the phenotype will change due to breeding and selection. That's going to be proportional to how heritable that trait is, and then how strong of a selection pressure you're applying. So here's an example I found from a tilapia breeding website. So basically, if this is like the phenotype of the parent population, so like it goes from small fish here to like large fish here, and this will be like the mean of the population. Most of the individuals are in the middle. And then the tilapia breeders, they pick this top like 10% of the fish, and then they breed those. What happens if the trait is heritable is that the offspring will be bigger than the parent population. So there's the mean of the offspring, and you can see it's significantly different from the previous generation, and that's because the trait's heritable and they selected the top biggest fish. A couple important terms when it comes to plant breeding are heirlooms and hybrids. And so heirlooms, they're called that because they were passed down in families. So it kind of refers to any kind of older variety that was like passed down across the years. And in order to qualify it as an heirloom, the variety should breed true, which means that the seeds from one fruit, like in this case, this is an heirloom tomato, the seeds from that fruit, if you planted them again, would make a tomato that looks similar or almost identical to the parent plant. And the way that heirlooms can do this is that they're inbred, so they don't have a lot of genetic diversity within the population of heirlooms. So that is a good thing in this case because the genetics are stabilized basically. So it's not really segregating for different traits from generation to generation. Like you're gonna keep getting the same tomato over and over again across the years. So that's a good thing. Plants can tolerate, I'll just note this here, plants can tolerate inbreeding a lot better than like animals or like humans can, partially because they are usually pretty self-fertile and they like self-pollinate a lot. So they are kind of adapted to doing that, whereas we need more genetic diversity. Okay, the other term is hybrid. And so here's the example here with hybrid corn, invented by George Scholl. And a hybrid, just like how Mendel used it, it's a cross between two inbred lines. But something that was noticed, and this has been shown to be true for a lot of different species, is that the hybrid tends to be like higher yielding and oftentimes bigger than either parent for some reason. So you can see here that the hybrid in the middle, the cobs are a lot longer and they have more seeds than the parents. And this is called heterosis, or also known as hybrid vigor. And there's some interesting genetic reasons for why that happens. But in terms of its usefulness, you get a more uniform phenotype. However, the hybrid does not breed true. So this is like an F1, just like in Mendel's experiments. So just like how you saw in the F2 generation, the genes that created this phenotype are going to start randomly being put into the offspring. So the F2 population is going to be a mixture of different phenotypes. So hybrid seed is basically made every year by crossing the two inbred parents. The seed companies will cross the inbred parents and sell the hybrid seed, and that's the standard for things like corn. So, after hearing all this, you probably want to do some plant breeding. So here are the steps to doing that. First of all, you need to pick a trait that you want to improve or change in your crop. I guess the step before that would be pick your crop, and then pick a trait that you want to improve or change. And then the most crucial step is finding parental material that has variation in that trait, because diversity is the raw material for breeding here. So if the trait that you're trying to breed for doesn't exist in the parent population, it's going to be hard to select for that, right? And then you want to make many crosses between those parental lines, and then select offspring based on the phenotypes that you like or that you're looking to change. And once you have something that you like, then the next step would be to try to stabilize the genetics by back crossing or selfing, if you can do that in your crop species. I will mention here that every crop and every plant is different on how people do breeding, and it depends on a lot of factors. So, for example, maybe it's hard to cross your plant for whatever reason. Maybe it's mostly like vegetatively propagated, and like potatoes, for example. Anyway, there's a lot of different reasons why, or like maybe you are growing trees and that are normally grafted instead of propagated using seeds. Anyway, but this is the basic outline for breeding. And also note that your variation must be heritable in order to select for it and see a change over time. So the variation does need to be genetic, but you don't actually need to know the underlying genes to do classical breeding. So anyone can do it. It takes a lot of time and a lot of seeds and a place to grow a lot of plants, but it's an exciting industry still. So variation is the raw material for breeding, but where does this variation come from? So of course natural variation is going to exist in like wild population, natural phenotypic variation, and genetic. But ultimately, all genetic variation originally arose from a mutation in the DNA sequence of the organism. And the other source of genetic variation is genetic recombination between different mutations and different alleles during sexual reproduction. So here's your two places you can get variation, and I'm going to talk about those more in the next section of this lecture. All right, we're finally going to talk about DNA and how genetics works at the molecular level. So this is kind of the broad overview of how you get from genotype to phenotype, and I'm going to go into this in more detail in the later slides. But essentially you have DNA, which codes for RNA, which codes for proteins, and the proteins are what give you the phenotype. Phenotype is also determined in a large way by gene expression, so which genes are on and off, and where and when that happens. And what are proteins? Proteins they make up the majority of things in the cell. So proteins are your enzymes, which make the different molecules and metabolites, and proteins also serve structural roles in the cell, and proteins also help orchestrate gene expression. And so some of the chemicals that are made by enzymes, proteins, are metabolites, pigments, colors, hormones, and many more. And so you have 4 basic letters of DNA that code for 22 amino acids that make up the proteins. And there's also a code for when the protein should start and stop. And a single change in the DNA can have large effects, and some changes have no effects. So, a bit of background terminology. DNA stands for deoxyribonucleic acid, and it is the hereditary material that stores the archive of genetic information for an organism. And genes are made of DNA, and gene is the functional unit of hereditary information. And so it carries that information from generation to generation. And the genes are packaged in chromosomes. And so one chromosome will contain many genes. It's the physical package for it. And then the genome is the term for all of the genes and all of the chromosomes for a given organism. And the genome and the genetic information is stored in the nucleus of the cell. So we looked at some nuclei during our labs, but inside there you can find the chromosomes. A specific location on a chromosome is sometimes called a locus. Plural is loci. And at a certain locus, you'll find a gene, and it's a given gene will be in the same place for all members of a species usually. Also, you'll see these chromosomes depicted as little X's a lot of the time, and that's because there are 2 copies of each chromosome in a diploid organism, like humans and a lot of other and a lot of plants too. And they pair up during when the cell's dividing, or for mitosis and meiosis. So they pair up together, and so the 2 chromosomes kind of make this like X shape as seen here. And the 2 chromosomes, they're going to have the same genes, the same loci, but they may have different alleles. So one copy comes from one parent, and the other copy comes from the other parent, and those parents may have different alleles for the genes. So DNA is the genetic code, and it has this double helix structure, and there are 4 chemical bases that make this up. And so we have adenine, thymine, guanine, and cytosine, and these are often just abbreviated by the first letter for the chemical. And the cool thing about these bases is that they can only pair up with their match. And so the chemical bases have shown that here. Basically, they kind of fit together like puzzle pieces, so that adenine always pairs with thymine, cytosine always pairs with guanine, and the cool thing about that is it facilitates replication. So when cells are dividing, the DNA gets unzipped here, and then each strand can be used as a template for a new strand of DNA, so it can duplicate itself. So how do you get from DNA to protein? This whole process is called gene expression. So each gene could be considered like a recipe for one protein. And then the way it gets there is the first step is a transcription. And so that is turning the DNA into RNA, and that's done by base pairing as well. So the RNA has similar bases and they match up with the DNA. And also this step is really highly regulated, so this is how genes can turn on and off, is like whether or not a gene is transcribed in the first place. And if a gene is on and it's transcribed, it makes the mRNA the messenger RNA, and that's basically a message that goes out of the nucleus into the cell, and then the proteins are manufactured by ribosomes there. And your final product is a protein, and it's going to have a specific sequence, and the amino acid sequence determines the structure and the function of the protein. And gene expression orchestrates development. And I like the term orchestrates because it is kind of like an orchestra. There's a lot of parts working all together to make this beautiful symphony that is a plant during development. So as we've learned many times throughout this course, plants are composed of numerous cell types that are all derived from the meristem tissue. And so the information needed to construct every cell type is in the genes of the organism in every cell. But all plant cells have the same genes. They grow by mitotic cell division, so that all the cells have identical sets of genes. And the way you get all these different cell types that you see, like xylem and phloem, is that different genes are turned on and off. So as the cell goes through the process of differentiation, It'll like turn on the xylem genes if it's going to be xylem and turn off some other genes, right? And that's orchestrated by regulating transcription and at certain times and places in the plant, and also influenced a lot by hormones. All right, let's look at the structure of a gene because it ends up being important when you start to think of what mutations do. So the structure of a gene is shown here, and basically the gene can be divided into different parts. So upstream of the gene at the beginning is something called a promoter, and that basically tells the RNA polymerase, which does transcription, where to start making the RNA. So it'll be like, okay, start here. And then the other important part of the gene is there's these regions called exons and introns. Essentially, exons are the coding sequence, so that's what's going to actually code for the protein, and then introns are non-coding. And the introns are actually going to be spliced out during transcription to give you the mature mRNA. And the mature mRNA will only have exons, so it only has the code for the protein, and that gets translated. There's also non-coding DNA in the genome overall. So on a chromosome, genes are interspersed in different locations across the chromosome, and in between you'll find regions of non-coding sequences. And sometimes this is called junk DNA, but it does serve some purposes. It can be structural and maybe regulatory. It could help turn genes on and off. A lot of the non-coding DNA is derived from these things called transposable elements, or TEs, and these are basically like remnants of viruses. And actually 85% of the maize genome is estimated to be derived from TEs. So the majority of the sequence here on a chromosome in corn is not coding for anything. It's just kind of these remnants from ancient viruses. So back to mutations. Mutations, the definition is any change that changes the sequence of DNA. And so these mutations may be spontaneous, and that usually arises from copying errors during DNA replication. It can also be caused by mutagens. So those are agents that mutate the sequence. Like radiation, for example, UV light, and there's a lot of chemicals that like bind to and damage DNA. And how this results in a mutation is damaged DNA. It's sensed by the cell, and the cell goes in and repairs that because DNA is really important for life, right? So the cell will try to repair any damaged DNA, and it can, because of the base pairing, it can use the sister strand of DNA to repair it, or sometimes it can even use the homologous chromosome as a template. But the repair process tends to be error prone, so this can result in new mutations. So what effects do mutations have on the phenotype? Okay, for one, a lot of mutations when they first appear are going to be recessive. So in a diploid organism, you have 2 copies of every gene. So you kind of have a backup copy. So if this mutation comes in this one gene and like messes up the gene, you kind of have a backup. So a lot of the times you won't, there might not be even an effect on the cell because this gene will continue making the good protein, and this one will maybe not be working, but it'll be fine. Of course, since it's recessive, if this organism that has this mutation were to inbreed, then some of the offspring would have 2 copies of the mutation. And so if that mutation was bad, then that would cause problems for that offspring. And in general, mutations can be good, bad, or neutral for the organism. Deleterious mutations are ones that have a negative effect, and the extreme example is like a lethal mutation, which messes things up so bad that if it's homozygous for that mutation, the organism will die. The majority of mutations are neutral and have no effect, and that's oftentimes because they're in non-coding regions of the genome, or they don't change the code enough to make a difference. And every once in a while, you'll get an advantageous mutation. So you'll get a change that somehow makes the protein better than it was before. Maybe it's more efficient at creating some metabolite or something. So, and that's, we want these in plant breeding advantageous mutations, and they're also going to be selected for by natural selection. And so, one of the reasons that the majority of mutations are neutral is that there's kind of this built-in redundancy of the genetic code. So, this is a codon table, and this is universal throughout all of life, which is pretty cool. And it shows you, like, for the different codons you can have, what amino acid will be specified by that code. And note, this table is written in RNA, and in RNA, the T's are changed to U's. So if the DNA says TTT, then the RNA will say UUU. And UUU here codes for this amino acid called phenylalanine. So, yeah, the definition of a codon is the 3-letter word here that specifies an amino acid. And the reason it's redundant is because you'll see that there are different codons that all specify the same amino acid. So, for example, here, all of these codons specify the amino acid called leucine. So, if you were to have a mutation that changed like this CUU sequence to CUC, you would still make leucine. So that mutation wouldn't really make a difference in the protein sequence. And this is known as a synonymous mutation. So the codon is changed, the DNA sequence is changed, but the amino acid is the same. You also can have non-synonymous mutations. So these do change the amino acid sequence. But this might not necessarily affect the protein structure or function. And that's because proteins are usually made of hundreds of amino acids. And not every amino acid is critical to the structure or function. Like, here's a drawing of a protein with different mutations like highlighted, and some are important and some are not, as it turns out. But non-synonymous mutations do have the potential to change either the protein structure or the protein function or both. So the types of mutations that create those non-synonymous and synonymous mutations are called point mutations. They're also called substitutions sometimes, and you'll also hear them referred to as single nucleotide polymorphisms, or SNPs. And this is what, like, those 23andMe or like Ancestry.com DNA kits test in humans, is they look for these single nucleotide polymorphisms. And then compare them to different ancestry and stuff to figure out where your ancestors came from. And so in a point mutation, one letter is basically just substituted for another letter. But it's going to keep the codons the same. Or it'll change the one codon that the mutation affects, right? So like here's a codon, and so this T got changed into a C. And as mentioned, these are usually neutral, but they could change the sequence if they're non-synonymous. Also, if the mutation occurs outside of a gene, it's probably not going to have an effect, but it could if it affects gene expression. So while a lot of the point mutations are neutral, these frameshift mutations are usually not so neutral. These are kind of the bad ones. And frameshift mutations are caused by either insertions or deletions of DNA bases. So in the diagram here, this extra C got inserted in the sequence. And the result of that is it kind of messes up the whole protein, the whole rest of the sequence. So there's like the codon. The codons are like 3-letter words, right? And that's like the reading frame is what it's called. So here we have our codon here, and then now there's this other one, and then all the other ones are going to be messed up after that mutation happened. And so these can be really problematic if they occur in the coding sequence of a gene, because you basically are messing up the whole protein. And I like this analogy here, so it shows the effect of a frame shift. Like, for example, if you had a sentence, the fat cat sat, and you deleted one letter and shifted it all over, then it's pretty much unreadable. So it's going to mess up the translation, and you're either going to get the wrong protein made, or a lot of times you'll get a shortened version of the protein, because it could introduce like a stop codon, and then the protein will just it'll end there. So this pretty much Yeah, it messes up proteins pretty bad. And these ones are going to cause more dramatic differences, usually not good ones, but every once in a while, maybe something you're looking for. Like if it knocks out a bad gene. And then I have to mention transposons, because these are those transposable elements that I mentioned make up a lot of the non-coding region. And the reason I want to mention them is because they can actually be mutagens themselves, because they basically encode proteins, the transposon sequence that encodes for a protein that cuts and pastes the transposon to a different part in the genome. So they're also been called jumping genes for this reason. So if the transposon lives here in a chromosome, it can jump from there to a new site in the genome. And the issue that causes is when it jumps, it's going to interrupt some other DNA sequence, which might be a gene. And so if it jumps in the middle of a gene, it's going to cause a frame shift and disrupt that gene's function. And these transposable elements were discovered by Barbara McClintock, pictured here, and she discovered them by looking at corn. She was looking at these little spots on some of these kernels of corn and wondering, how was that genetically encoded. And she was actually, when she first presented her results in the 50s, people weren't couldn't understand her. She was too far ahead of her time, basically, and they thought that there was no way there could be elements of the genome that moved around. But it turned out she was right, and this is responsible for a lot of this kind of variation that you see. All right, so how do these mutations change the phenotype? So, as I mentioned before, a lot of proteins are enzymes, and plants, I think, are some of the best organic chemists in the world. So they can make a lot of cool compounds using their enzymes. And so their enzymes are kind of like the little molecular chemists that live in cells and they synthesize chemicals for the plant. So enzymes are proteins. Here's a diagram. This shows how anthocyanins are made. So it's the anthocyanin synthesis pathway. And anthocyanins are purple and pink pigments in plants. And so it's definitely hard to read these diagrams, but the starting material is phenylalanine, which is an amino acid. And then each one of these arrows is a synthesis step. And each one of these blue symbols is a gene. So this gene converts phenylalanine to the cinnamonic acid, and then that keeps getting converted through 1, 2, 3, 4, 5, 6, 7, 8 steps, and you get some anthocyanins, some pigmented molecules. And yeah, so each transformation of the chemical, that is done by an enzyme, which is encoded by a gene. So let's say you have a purple plant. If there was a mutation in one of these genes that synthesized that purple molecule, this gene, for example, got mutated, then the plant would not be purple anymore. It would be probably white. So, and another way to kind of illustrate that is this is supposed to be a molecule. I'm just drawing it as a circle. So these molecules here are not earlier in the pathway, these don't have any pigment, they're not colored. So the enzyme converts that to something that's purple. And the enzyme's coded by DNA. So this is the basis for Mendel's peas. If you knock out that gene, then you don't have the enzyme, then you don't have purple. That's going to give you white flowers, for example. And then there is also some large-scale genetic variation. I won't really get into it very much, but there's something called copy number variation, which is excuse me. It's very popular in plants. They really like to duplicate their genes for some reason, and and this can happen during replication or other events like transposon action. But if you have an area, you'll have an area of the chromosome, a whole area with a bunch of genes that just gets duplicated, and then now you have 2 copies of all of those genes. And yeah, normally that would be pretty harmful, in humans, but plants are able to handle that better. They're more equipped, and they actually use this to their advantage to evolve new forms of genes. A lot of the genes that synthesize different things have evolved that way. There's also examples of translocation where like one part of a chromosome will get moved to a totally different chromosome. You'll also sometimes see extra copies of chromosomes and even whole genome duplications. Speaking of whole genome duplications, let's talk about polyploidy, or as I like to say, how many genomes do you need? So, ploidy refers to the number of chromosomes per cell, and that is often abbreviated with a lowercase n. So, the lowest number is haploid, and so this is the number of chromosomes that gametes have, like eggs and sperm cells, and it's usually half of the number of the parents. And then the diploid, and also there, some fungi and stuff and bacteria are haploid their whole life. Diploid is 2 copies of each gene, and that's pretty traditional for somatic cells, body cells, and a lot of animals and plants. But you do get polyploids in plants, and they have interesting effects on the phenotype. So 3N would be triploid, and these are usually sterile because if you have 3 chromosomes, like trying to come together during meiosis and split in the right amounts doesn't really happen accurately. But this can sometimes be advantageous because if it's sterile, it usually doesn't produce seeds. For example, like the seedless watermelon is actually a triploid. But it can't be propagated by seeds, right? And then 4N is tetraploid, and a famous example of that's a potato. 6N is hexaploid, and wheat is the poster child for that. And then 8N is octoploid, and that's actually the modern strawberry that you all eat and enjoy is an octoploid. One interesting consequence of polyploidy in plants is that it tends to make the plant, like the fruits and the plant cells, bigger, like larger, which is great if you're breeding for big strawberries, right? Or big potatoes. On the other hand, though, the more copies of each gene that you have, the trickier breeding's going to be. So, like, to try to, like, stabilize the genetics when you have 8 copies, it's going to take a lot longer, and it's going to be trickier. And you also, typically, you can't cross plants with different ploidy levels. So, if you try to cross, like, this octoploid strawberry and this diploid one, it wouldn't be able to make a viable zygote or embryo because like there's not the right amount of chromosomes. They can't do mitosis properly and stuff, so it's not going to work. So it can make breeding difficult. And yeah, the polyploidy can arise also from hybridization. Sometimes 2 different species can cause that, or also some whole genome duplication events. Alright, and this is my last slide for this section. I just wanted to talk a little bit about linkage and crossing over because it's another important source of novel genetic combinations. So crossing over basically happens during meiosis, and it ends up, so if 2 genes are linked, it's because they're on the same chromosome, right? So in this example here, they have like A and B. So A and B are 2 genes on the same chromosome, on different arms in this case. And so you have one parent has like little a, little b, and then the other parent has big A and big B. So during meiosis, you have these events called crossovers where basically like one of the arms crosses over to the other one and they end up getting switched when the cells divide. So if you remember from reproduction, meiosis, you have like 1 copy of each, one from the mom, one from the dad. Those duplicate, and then they come together, crossing over happens, and they split off into 2. And then those 2 get split off again into haploid gametes. And that's shown here also. I just have to do with my fingers to remember how it works. Okay, so anyway, crossing over. results in genetic recombination, though, new combinations of genes. So before, on this chromosome here, you have in the gametes, you'll have chromosome copies that have the same as the parent, and then you'll have these new recombinants that have a different combination of of alleles for these genes. And this is a major source of genetic diversity for breeding, because new combinations can create new phenotypes. However, the downside is it can be a barrier to stabilizing phenotypes. So if you have some line that's really perfect, and there's some linkage between traits that you want to keep together, this can break that linkage, if that makes sense. Okay. There's a lot more to say on that topic, but I think this will do for now. Thank you for listening.